Let's get started. Good afternoon, everyone. My name is Günther Wagner. I'm organizing the Perinatalology Research Branch lecture series on the developmental evolution of female reproduction. And it's my particular pleasure today to invite, welcome our first outside speaker, Professor Vincent Lynch from the University at Buffalo. And he's going to talk to us today about the evolutionary regulation of embryo implantation. Vinny, take it from here.
Thank you for inviting me, Günther. Günther requested that I put this first slide in, so just everyone can see what the upcoming lectures are on. As Günther mentioned, I'm going to be speaking today about some evolutionary aspects involving the regulation of embryo implantation. And before I get started, I just want to point out that I tend to get excited when I talk about this stuff and talk faster and faster and faster. So I don't mind being interrupted when I'm giving a talk. Chances are if you don't understand something, then someone else doesn't, and it's probably because I've just misspoken. So if something doesn't make sense, feel free to stop me and ask for clarification. I know that this is the third of these, and Günther has probably already given some background information, but in case you haven't thought about this in a while, mammalian reproduction tends to be different than other mammals or other animals in lots of different ways. So in this phylogeny, I'm showing you how the tetrapods are related to each other. And these are sort of the extant mammals, right? So we have mammals like us and elephants, mammals like marsupials, and then mammals like the egg-laying monotremes. So even though monotremes lay a shelled egg, they have a very short period of pregnancy. So for about 2 weeks, the embryo, while it's developing, hangs out in the uterus. And during this period of egg retention, most of the nutrition for fetal development is coming from mom, actually coming from the uterus, rather than the yolk source. So platypus and echidna, they do have yolk. They just don't have a lot of it. So this period of egg retention, the uterus is sort of dirty. It's sloughing off a lot of mucopolysaccharides and lots of other vitamins and minerals, which are taken up by a very simple placenta on the underside of the shell. So they're able to get away with this because the shell is very porous. It's not like a chicken shell, right? Like a chicken egg, you can crack it. Not that you would do this to a platypus egg, but you can't really crack a platypus egg. It's sort of leathery, like a reptile egg. And it's very porous, so stuff can pass through that eggshell and then be taken up on the underside by a very simple two-layer placenta. So platypus and echidna have this sort of equality that they're in between being egg layers and live bearers, right? So they lay eggs, but they have some of the aspects of live bearing, like Therian mammals. They don't have to worry about some of the challenges that Therian mammals have to worry about, though, because the embryo itself, even though it's sort of expressing a bunch of antigen that should be recognized by the maternal immune system as foreign, it's sort of cloaked because it still has the shell there, and the shell is immunologically inert. So they get pregnant, but they don't have a lot of the challenges that go along with pregnancy in Therian mammals, like maternal recognition of pregnancy, because there's a shell there that gets in the way. That shell disappears in the common ancestor of Therian mammals, which are placental mammals and marsupials. So in this lineage is when we get the evolution of full viviparity and an elaboration of placentation. So we no longer have a mineralized eggshell in Therian mammals, even though we have part of the eggshell membrane that the embryo or the egg still has to hatch from. And we have completely lost yolk. The yolk sac is still there. It performs different functions in Therians and eutherians, however. If you're a eutherian mammal like us, the yolk sac has has functions very early on in development. Those seem to disappear. In marsupials, the yolk sac actually turns into the placenta. So the placenta in marsupials is derived from the yolk sac, while the placenta in eutherian mammals like us is derived from the placenta in eutherian mammals like us is derived from the chorion of the atlantois. And then finally, the really interesting things happen in eutherian mammals. So eutherian mammals are sometimes called placental mammals, but that's not really true because marsupials and monotremes have placentas. So in this lineage is when you get things like the evolution of implantation, maternal recognition of pregnancy, immune tolerance. So the maternal immune system doesn't recognize the fetus as non-self and mount an immune response against it. In fact, like you all know, it does just the opposite, right? The maternal immune system is essential for promoting implantation, promoting pregnancy, and maintaining it. And you also get prolonged gestation. So so when we talk about maternal recognition of pregnancy in these species, we don't actually obviously mean like in humans where you can say, oh, I've missed a period, I know that I'm pregnant. There's a physiological response in eutherian mammals to the presence of the conceptus that doesn't happen in most marsupials. It happens in a couple, but not most. And that's most noticeable in that in eutherian mammals, when they get pregnant, the reproductive cycle stops until after pregnancy is over. That doesn't happen in marsupials. So in most marsupials, the reproductive cycle keeps going, regardless of whether birth happens. So you could get this weird situation like in wallabies, for example, where they have two uterine horns and they're pregnant from different reproductive cycles in both uterine horns, so they're constantly pregnant. And that's because the reproductive cycle isn't interrupted by pregnancy like it is in eutherian mammals. In order to be able to pull that off, you have to have some way for the mother to recognize that she's pregnant, and you need some way to have gestation last longer than the length of a single reproductive cycle. So that's why reproductive cycles in marsupials are short, 2 to 4 weeks, and reproductive cycles in eutherians can be really long, just under 2 years if you're an elephant. Many eutherian mammals have also evolved this thing called implantation. And I'm sure I don't have to describe what implantation is, but just in case, right? It's the embedding of the embryo directly into maternal tissue. So that is, as far as I'm aware, Hunter might know if that's not true, the only species in which implantation occurs are eutherian mammals, even though pregnancy happens in other things, right? So there are reptiles that can get pregnant, and there are sharks that can get pregnant, and there are fish that can get pregnant. But in all of those species, implantation doesn't happen. So the embryo might come up to the wall of the uterus and stop there, but direct implantation of embryological tissue into maternal tissue appears to be a unique aspect of some eutherian mammals. And that's just within the eutherians proper. Within primates and humans, things evolve even more, right? So humans and many other primates, mostly old-world monkeys in this context, have a lot of these eutherian pregnancy traits, but they also have some derived traits that are unique to themselves. So humans and a few other animals menstruate, for example. We have really deeply invasive implantation, this so-called interstitial implantation, in which the blastocyst comes to be completely enveloped within the endometrium. I mean, it's not quite clear actually if the blastocyst is burrowing into the endometrium or the endometrium is growing out and sort of being encapsulating the blastocyst. But either way, the blastocyst ends up almost completely encased within the endometrium. Humans and other old world monkeys have very, very highly invasive placentas. We don't have the standard parturition signal that most other eutherian mammals have, which is systemic progesterone withdrawal. Whatever it is, we're not quite sure. It's probably withdrawal of tolerance, but sort of the signal that initiates withdrawal of tolerance is not known. And humans seem to be particularly susceptible to adverse pregnancy outcomes, especially infertility, preeclampsia, and preterm birth. And being susceptible to these adverse pregnancy outcomes is likely because of these other things, right? So if you don't evolve an invasive placenta, then you probably aren't going to have preeclampsia. So all those other species of eutherian mammals which don't have invasive placentas don't have preeclampsia. As far as I'm aware, there's not a preeclamptic whale because whales don't have deeply invasive placentas. So what we want to know is how did all this evolve, right? So how do we get to this point where we have all of these traits? What we've been doing in the lab actually since I was in Gunter's lab, so since I was a grad student, is something that we call ancestral transcriptome reconstruction, which is pretty simple, right? So what we do is we assemble a collection of transcriptomes from basically anything that we can get our hands on. At the moment, this includes 24 eutherian mammals, 3 marsupials, a platypus, 3 birds, and 8 lizards, including lizards that are both viviparous and oviparous. So these are lizards that give live birth and that lay eggs. And we collect the endometrium from early to mid-stage with the realization that pregnancy lasts different lengths in different species, right? So you can't just say collect it from day 42 because some species might not make it to day 42. And in other species, day 42 may be very, very early in pregnancy. What we try to do is say, okay, first or second trimester samples from whatever species we happen to get. And then sequence the transcriptomes using RNA-seq. And then what we've been doing is using a model-based method to classify genes as expressed or not, right? So the dirty secret from RNA-seq data is that, if you generate 100 million reads, chances are every gene in the genome is going to appear in your RNA-seq data as expressed, even if it's expressed at expression levels, which are extremely low, right? So is a gene that has an extremely low expression level, do we call that expressed or not? So we try to get around this by just saying, well, let's categorize things into 2 states, expressed or not. So here as well, I'll just give a brief digression into how we do that. So how do we turn and why do we turn these continuous gene expression quantifications into binary gene expression states? And this actually goes back to an old observation, at least from 2011 and 2012, that if you look at gene expression data from both bulk RNA-seq, not single-cell RNA-seq, and even microarray data, gene expression values are usually bimodal. And you can classify these bimodal expression values into high and low gene expression categories. So in this histogram, what I'm showing is bulk RNA-seq data from human endometrial stromal cells. On the x-axis is the expression level in transcripts per million, which is just the molar ratio of that transcript, and it's logged. And on the y-axis is the density. And you can see that if you look at this data, there clearly looks like there's 2 distributions. There's a low distribution corresponding to low expression levels, and another distribution corresponding to high expression levels. So you can actually do math and decompose this single distribution into 2 distributions, and they look relatively Gaussian, so that's what we did. We decomposed these 2 expression states. We can decompose this gene expression levels into 2 gene expression states that mostly correspond to low expression and high expression. And then you can plot over that the empirical cumulative frequency distribution plot. And around TPM of 1 or 2, you can find that 50% of the genes will be expressed above that, and 50% of the genes will be expressed below that. So here is where you have to add a sort of a preference. So we call a gene that's expressed at TPM greater than 2 expressed, so state 1. And genes that are expressed at TPM less than 2 not expressed, or state 0. But you can see that there's some gray area in here where the 2 distributions overlap. So you could move that cutoff down or up depending on your preference. So if you want to make sure that you have, that you're calling every gene that might be expressed, maybe you want to move your expression cutoff down. And if you want to be really sure that something that you call is expressed, maybe you want to move your expression cutoff up. We found that sort of the sweet spot for this kind of data is a TPM of around 1 or 2. Practically it doesn't matter when you do the analyses, but it looks like the TPM of 2 is most efficient for these kinds of studies. The nice thing is that these expression levels, once you binaryize them, look like they correspond to sort of functionally relevant gene sets in the genome. So if you look at different markers, different genetic or genomic markers for promoters, enhancers, and regions of open chromatin, and then you look at the different gene expression categories, so let's say the genes that we classify as expressed at TPM greater than 2, not expressed, so these are genes that have, in green, expression values between 0.01 and 1, so expressed very, very lowly, or not expressed at all in black. You can see that if we call a gene as expressed with a TPM of greater than or equal to 2, it tends to have the strongest promoter mark. It tends to have the greatest enhancer marks. It tends to be in regions of open chromatin, while the more lowly expressed sets tend to not have open enhancer, or tend to not have active promoter marks, not have active enhancer marks. It's a little wishy-washy for open chromatin. But if you then look at RNA pol2 ChIP-seq data, so this is ChIP-seq data for the enzyme which is actually transcribing DNA, you can see that genes with TPM of greater than or equal to 2 have the most evidence for active transcription. Genes with TPM of less than 2, so in the green, tend to not have evidence of active transcription. So TPM2 looks like it captures most aspects of being expressed or not. The other thing that's nice for doing this is that we're comparing data from lots and lots of different species generated over many different many different experiments using different sequencing technologies. And at the risk of summoning the batch effects gods, we want to make sure that whatever we're doing, when we analyze our data, we want to minimize the technical effects of sequencing as much as we can. So what I'm showing here is an MDS plot, which is kind of like a PCA plot. It's a way of looking at the contribution of variants to gene expression clustering. For all of the species, if we encode gene expression in the quantitative way as a transcripts per million, and what you can see is that there doesn't seem to be clustering that makes sense. So here the species are colored by their lineage. So red means eutherian mammal, blue is platypus, dark blue opossum, wallaby, dunart, so those are the marsupials, and black are the non-mammals. And you can see that they don't seem to form distinct clusters. Naively, one might expect that species that are most closely related to each other would have more similar gene expression states, so they would form clusters. So maybe we'd get a cluster of primates down here and a cluster of reptiles over here. But that doesn't happen when we cluster genes using the transcripts per million values, so the quantitative gene expression values. But when we transform those values into the binary encoding, into zeros and ones, then we do reveal clustering by species relatedness, which is what one expects if you're removing noise and revealing signals. So now you get a cluster of primates down here, a cluster of eutherians in red, and a cluster of birds and reptiles in black. So binary encoding in this way reflects natural gene expression states and reduces the potentially many sources of noise that are generated in RNA-seq data and sort of allow us to do everything that comes after. Right? So we classify all of our genes as expressed or not, and then we do the evolutionary thing. So what we can next do is infer ancestral gene expression states. You can do that lots of ways. So we used an empirical Bayesian method implemented in a software tool called IQ-Tree. And what you give IQ-Tree is your matrix of gene expression states in zeros and ones. You give it your species phylogeny. And then you have it infer a model of evolution, which is just a model for how you expect gene expression to evolve. And then you could use these methods to reconstruct ancestral gene expression states and then trace changes in gene expression. So trace changes in zeros and ones across the phylogeny. Here's a little cartoon example of what that might look like if you're not an evolutionary biologist and don't really think about these things often. So imagine you have some gene A, and you find that it's not expressed in any of the non-eutherian mammals, but it is expressed in all of the eutherian mammals. So you get state 0 here and state 1 here. There are potentially many evolutionary scenarios that can explain these data. The simplest is that there was one change from 0 to 1 in the common ancestor of all the eutherian mammals because they have the gene expressed. Well, nobody else does. And this is implicitly probably what you did when you looked at these data. An alternative model, though, is that the gene is ancestrally expressed and evolved to not be expressed 1, 2, 3, 4 times. Most parsimoniously, this doesn't make a lot of sense, right? Because this requires 4 evolutionary events. Well, this explanation requires only 1 evolutionary event. So we tend to prefer the reconstruction that requires the fewest number of events. So this is a parsimony-based method, which is not that different from a maximum likelihood-based method. The nice thing about likelihood, which is what we are using now, is that it allows us to also infer how certain we are that this gene expression change occurred here. So we get to say the method reconstructs a 0 to 1 change along this branch. And then along with that, we get an estimate of how certain we are between 0 and 1. That gives us the probability that the reconstruction there is correct. So then we can do that for every gene, for every branch, and identify all of the changes in zeros and ones throughout the phylogeny. And for each of those, get an estimate of certainty or uncertainty for whether that reconstruction is correct. When you do that for the whole tree, what you find is that gene expression change is really common. So hundreds of genes gained, showing a plus, or lost endometrial expression during the evolution of a million pregnancies. So here are just some lineages and the numbers associated with those. So these are lineages going backward from human. So this branch, this branch, this branch, this one, this one, and this one. And we're only considering, we exclude all of the gene expression changes that have uncertain estimates. So an estimate that's less than 80%. So you can see that lots of gene expression changes occur throughout lots of different species, but also throughout the ancestral human lineage. So for example, in the common ancestor of eutherians, when implantation, maternal recognition of pregnancy, maternal fetal tolerance, recognition of the fetal allograft, so when all that evolves, there's about 1,000 genes which went from not being expressed to being expressed. So they evolved to be expressed. And there's about 700 genes which evolved the opposite direction, evolved to not be expressed. Right? So these are genes which are present in the genomes of all these other species. They're just not expressed in the uterus during pregnancy in those species. And what I find really interesting is that the magnitude of the effect of the magnitude of the gene expression changes associated with the origin of pregnancy is about, you gain about 1,000 genes, and you lose the expression of about 700 genes. It's larger in humans. So since the last common ancestor of humans with the old world monkeys, there have been 1,500 gene expression gains and 1,285 gene expression losses, which suggests that we might think that the origin of pregnancy is a major event, but it also looks like there's big events going on in humans and probably other species as well. And you can infer this by looking at the tree, just by looking at how long these branches are. See how long the human branch is? That means there's a lot of change happening on that branch. Meanwhile, the origin of pregnancy branch is this one, so it's relatively short. So there's been a lot of change happening in the human lineage compared to the ancestral ones. And this seems to be true for a lot of the extant species, like how long the mouse branch is and the dog branch is and the goat branch is. So then what we did next was take all of the extant gene expression data and the reconstructed ancestors for each one of these nodes. We generated a big matrix of zeros and ones for all the extant data and all the reconstructions. And then we try to cluster them using various different ways. So again, this is an MDS plot, a multidimensional scaling plot. Conceptually, it's similar to what's going on with the PCA plot. It's difficult to use PCA on binary data. You technically can't do it. So MDS allows you to cluster binary data. Again, this is trying to look at how similar or different things are from each other, and in a relatively unbiased way based on the variance in the gene expression levels, in this case, zeros and ones. So what you can see is that there are nice clusters in the data. So this is the MDS plot. These are the clusters. These are clusters determined with k-means clustering. So we don't actually assign a gene, we don't actually assign a species to a cluster. We let the clusters find themselves through k-means clustering. And you can see that you end up with a cluster over here, which is basically everything that lays an egg or is a bird and reptile. So duck, Chalcides is a lizard, Cyphos is a lizard, chicken is a lizard, platypus, right? So egg-laying mammal. Platypus doesn't cluster with the other mammals. Platypus is an egg layer, and it clusters with the other things that lay eggs. So then you can further break these clusters down. So you get two clusters here. These are species that have non-invasive epithelial chorial placentas, right? So things like buffaloes and sheep and goats and cows and possums, right? So again, possum is, it has a different kind of placenta than the eutherian mammal, right? The eutherian mammal placenta is derived from the chorion of the elantois, while the possum placenta is derived from the yolk sac, but the possum gene expression data in the endometrium clusters with the other species that have non-invasive placentas. And right, this is the endometrial gene expression data, not the placental gene expression data. So when we collect these tissues, we remove the placenta. So we can predict endometrial gene expression. We can predict placental invasiveness based on gene expression in the endometrium. So we get non-invasive clusters. This cluster corresponds to those species that have a hemocorial placenta, so mouse, armadillo, rabbit, squirrel, hamster, and bat. And then we get a cluster at the bottom, which corresponds to species that have a hemocorial placenta, like humans and macaques and baboons, but it's different, right? So this cluster down here is hemocorial, but the primates seem to be clustering distinct from the other species that have hemocorial placentas. So gene expression in the uterus can predict placental invasiveness, but it also seems to differentiate humans and other primates from the other eutherian mammals. And this trick also seems to work for the deep nodes, right? So the ancestral nodes, like the ancestral primate, based on its gene expression pattern, node 67, the ancestor of the primate is here, it clusters with the other primates. So we can predict ancestral gene expression states of the endometrium based on their clustering. So we could probably say with the kind of certainty that we can do with these methods that the ancestor of primates probably had a gene expression state that's similar to those kinds of things with hemocorial placentas. And we can move deep into the tree and say that, well, the common ancestor of eutherian mammals has a gene expression state which is similar to other species with hemocorial placentas. Meanwhile, basically everything that lays an egg or is a bird and reptile has a gene expression state which is similar to things that lay eggs and eggs. And what I thought was most interesting when I noticed this, when I was looking at these data, is that primates clearly are different here, right? So I sort of expected humans, macaques, and baboons would be clustering with the other species that have hemocorial placentas, but they're clearly standing out, which is an indication that gene expression in the endometrium during pregnancy in primates is different than gene expression in the other eutherian mammals, even the other eutherian mammals with hemocorial placentas, which is probably associated with unique gene expression changes that have occurred in the endometrium in these species. If we look to see how many of those there are, for example, if we look just in the human lineage, there are 923 genes that have evolved to be expressed in the endometrium during pregnancy in the human lineage with a probability of 1. So there's no uncertainty here. So that's 1,000-ish genes which have evolved to be uniquely expressed in the human endometrium during pregnancy. And ideally, we'd like to know what these things are. It's, of course, hard to do those kinds of experiments, but we can try to infer it in lots of different ways. So if we want to know what are the consequences of these gene expression changes for human pregnancy, what we can do is go back to other kinds of data sets and look to see what genes, what pathways, what gene ontologies these genes are enriched in. So if we take those 1,000 genes that have human-specific, we take those 1,000 genes that are expressed in the human uterus during pregnancy, and we look to see what gene ontology terms and disease ontologies and pathways are enriched in, you get these. So if you're not used to looking at one of these, this is just a word cloud. It's a way of showing all of the same information that would be included in a table, just in a more visual way. Right? So the size of the term here is proportional to the significance of that term. So if we look to see of those 1,000 genes, what are they enriched in? A lot of these genes are enriched in genes associated with preterm birth. Very much fewer are associated with leukocyte activation involved in inflammatory response. So you don't have to see all of the words here to see some standouts, right? So genes that have evolved to be expressed in the human lineage are enriched in genes associated with preterm birth, but so are genes that have evolved to be lost, whose expression is lost in the human lineage. So genes that change expression in humans, both gain and lose expression, are associated with preterm birth, They're associated with general things that we might associate with pregnancy, like pregnancy, first trimester pregnancy, abortion, preeclampsia, graft versus host disease, right? Because the fetus is nothing but an allograft, so it might make a lot of sense that you get a lot of autoimmune things like graft versus host disease and organ transplantation. Again, you get a lot of autoimmune things because pregnancy is an immune process, so you get a lot of association with autoimmune diseases and asthma and rhinitis and immune system diseases. Then some things which don't necessarily make a lot of sense, like serotonin signaling. There's a lot of genes associated with serotonin signaling. CAMP signaling makes sense because we know that CAMP-mediated signaling plays an important role in pregnancy, in particular in decidualization. So it looks like those 1,000 genes, which evolved to be expressed in the human uterus during pregnancy, are probably playing important roles in pregnancy-related phenotypes. These are obviously difficult to get at more mechanistically because it's difficult to study human pregnancy mechanistically. So I have no doubt that I don't need to tell you that the human uterus is a busy place. There are lots of different cell types at the maternal-fetal interface during pregnancy. And what we next wanted to know was, are these human recruited genes, these 1,000 genes, which evolved to be expressed in the human uterus during pregnancy, are they enriched in any specific cell type, or are they just expressed in lots and lots of different cell types? So what we did was we grabbed the Vento-Tormo et al. Single-cell sequencing data set from the human first trimester decidua. This is just a UMAP showing the different clustering of cells from their data. And then we took those 1,000 genes, which evolved to be expressed in the human uterus, and tested whether their expression was enriched in any of these cell populations. And what we found is that indeed there was one major cell type, well, a couple, but the one major cell type stands out. So those human recruited genes are almost all enriched very significantly in decidual stromal cells. So this population here. So there's 1, 2, 3, 4, 5 different populations of decidual stromal cells in which the expression of human recruited genes are enriched. Those genes are also enriched in plasmacytes, endothelial cells, and dendritic cells, and they're not enriched in any other cell type. So the human recruited genes predominantly seem to play a role in gene expression evolution and endometrial stromal cells, which is great because I'm an endometrial stromal cell partisan. I tend to think that that cell type is the important one. Lots of other cell types are important, but evolutionarily and probably for various kinds of adverse pregnancy outcomes, I think that the endometrial stromal cell is playing a central role in mediating those things. So that's, again, just list building, right? So we've identified a bunch of genes which evolved to be expressed in the uterus. In humans, it looks like those genes are preferentially expressed in endometrial stromal cells. So what are they, and what are they doing there? For the next story, I'm gonna focus on just one of these and try to give you a little vignette about how we can use these kinds of evolutionary data to hopefully infer some kind of new biology that tells us something about human pregnancy, and in this case, probably human implantation and maternal-fetal communication. So lots of these terms made sense, but the serotonin, right? So these human recruited genes, which are expressed in the human uterus during pregnancy, were enriched in serotonin pathways and CAMP-mediated signaling. And that didn't make any sense to us. So we decided to go after those genes and see what role they may have in endometrial stromal cells. So we wanted to answer the question, what's going on with these serotonin signaling pathways? Among the genes, which are annotated with serotonin signaling roles, is the serotonin receptor HTR2B. There's a whole bunch of different serotonin receptors. This is just one of the serotonin receptors. And in this phylogeny, what I'm doing is showing you the expression state of that serotonin receptor gene, HTR2B, and all the different species and its reconstructions. So it's shown in gray if it's not expressed, and it's shown in black if it's expressed. And it's a pie chart, so the more black, the more certain we are of its expression. So what you can see is that it's not really the case that only humans express the serotonin receptor HTR2B. It's expressed in some of the lizards, it's expressed in chicken, it's expressed in Dunart, it's expressed in mouse, squirrel, baboon, pig-tailed macaque, human, armadillo, and platypus. But most species don't express it. And when you look at the evolutionary history, what you see is that it evolved from ancestrally not being expressed to being expressed in these species, independently from chicken, independently from Dunart, independently from mouse, and independently from the primates. So what it looks like it happened is that there are multiple species that independently evolved to have the serotonin receptor HTR2B expressed in their uterus, including humans and, interestingly, mouse. And when we look to see which of those cell types from the Ventotormo data express the serotonin receptor HTR2B, its expression is almost entirely restricted to this population, which are the endometrial stromal fibroblast populations. So it's, there's a little bit of expression in some of these macrophage, but most of the expression is almost entirely restricted to the decidual stromal cell populations. I said before that there's a bunch of different serotonin receptors. These are all of those serotonin receptors. And as far as we can tell, at least in human endometrial stromal fibroblasts and human decidual stromal cells, if we look at the RNA-seq data for DSC in gray and ESF in black, HTR2B is the only one that is expressed. So none of the other serotonin receptors are expressed. And if we look at GTEx data for a whole bunch of other tissues, you can see that the HTR2B receptor is most highly expressed in the uterus and the adrenal gland. So it's rank order 2 of the uterus. So it looks like, so if you go and look at the adrenal gland data and the uterine data, it looks like this serotonin receptor is very highly specific for uterus and adrenal gland. So what's it doing in stromal cells? As far as we know, there wasn't a demonstrated role for serotonin in stromal cells, let alone at the maternal fetal interface. So what's the serotonin receptor doing in endometrial stromal cells, and why might it be different in humans? So what we did was we took those single-cell sequencing data from Ventotormo. We did a pseudo-time differentiation trajectory of the endometrial stromal cell population. And this is just looking at the differentiation of endometrial stromal cells into decidual stromal cells. So these are the endometrial stromal cells. This is the perivascular population, which is probably representing the mesenchymal stem cell-like population from which ESFs derive. And it looks like endometrial stromal fibroblasts differentiate into one of two kinds of decidual stromal cells. Right? So you get a differentiation trajectory this way, leading to populations 3, 1, and 2, and a different differentiation this way, leading to populations 4 and 5. There's a branch point. And if you look to see which of these cells express the serotonin receptor, it's many of them. Notice that it's mostly the decidual stromal cells, not the endometrial stromal cells, and not the stem cell-like precursor of the stromal fibroblasts. And it's mostly lineage 1 cells. So not all stromal cells express HTR2B. It looks like it's a subpopulation that express HTR2B. So then what we can do is we can compare the gene expression signatures of the cells that express HTR2B to the cells that do not express HTR2B. So what we can do is, for these cells in red, compare them to the cells in blue and say which genes are co-expressed with HTR2B in these cells and which are not co-expressed with HTR2B. And that's what this little volcano plot is showing. So it looks like there are 84 genes that are co-expressed with HTR2B and these HTR2B positive cells compared to the HTR2B negative cells, which isn't very many genes, right? There are 84 genes that are there, and there's what, 10, 12,000 genes expressed in these cell types. So it's just a small number of genes which are differentially expressed. Although some of those genes are probably important. IGF-1 and 3 is probably really important. IL-15 is a signaling molecule which plays an important role in immune cell recruitment into the uterus during pregnancy. So it seems like there's important genes that are co-expressed with HTR2B. So if you take those differentially expressed genes and then do an enrichment analysis, these are the results that you get, right? So genes that are differentially expressed between HTR2B positive and negative cells, and there's 194 of them total, are enriched in pathways and disease ontologies that are related to different kinds of adverse pregnancy outcomes. So you get PROM, chorioamniotis, premature birth, again, rupture here is referring to membranes, endometriosis, spontaneous abortion. So lots of diseases that we associate with sort of human adverse pregnancy outcomes. And then there's a bunch of pathways which are also related to these things. So prostaglandin synthesis and regulation is probably playing an important role both in recognition of pregnancy and the initiation of labor and delivery. Obviously, smooth muscle contraction plays an important role in labor, myometrial relaxation and contraction. And the aryl hydrocarbon receptor is probably playing an important role in maternal recognition of pregnancy. And then you also get some interesting terms for different reasons. So you get things like neoplasm invasiveness and neoplasm metastasis. There are lots of reasons that these are interesting, but you can sort of think of the placenta as a kind of metastatic cancer that invades into maternal tissues. So part of that invasion process is probably mechanistically and evolutionarily derived from blastocyst implantation. So we think that maybe blastocyst implantation evolves early in the evolution of pregnancy, and those pathways which mediate implantation of the blastocyst into maternal tissues can be co-opted if they're inappropriately expressed in cancer cells to drive neoplasm metastasis. So this data is all building to suggest that the HTR2B gene is playing a role in decidual stromal cells, and we'd like to know what that role is. So one of the first things that we did was just look to see whether the gene, whether the locus in the genome has hallmarks of a progesterone responsive gene and a gene that might be expressed, actively expressed. So you can see that it has a promoter mark, it has lots of enhancers nearby it. It's in a region of open chromatin and has some open chromatin nearby. It's bound by pol2, so it's actively transcribed at a relatively high level for the pol2 ChIP-seq to be able to identify it. And if you look at ChIP-seq data for various transcription factors, which you know play important roles in the endometrial stromal cells, like the progesterone receptor, FOXO1, FOS, GATA, COOP-TF, or NR2F2, you can see that there's a whole bunch of transcription factor binding sites, experimentally verified transcription factor binding sites nearby the HTR2B locus. And finally, if you look at chromatin conformation data and look to see where are regions to which the HTR2B promoter are looping, which are shown by these, it's looping to lots of regions of the genome that are open chromatin, have enhancer marks, and have transcription factor binding sites. So all of these data suggest that HTR2B is actively expressed in stromal cells. And if you look at the RNA-seq data, it looks like it's pretty highly expressed in endometrial stromal fibroblasts and transcriptionally downregulated during decidualization into decidual stromal cells. But even though it's downregulated, its expression level is still very high. So a transcript per million expression level 100 is a very highly expressed gene in this cell type. So then we grab some data that already existed to look at gene expression throughout the menstrual cycle and throughout gestation, and that's these data. So here you're looking at gene expression of HTR2B in different phases of the menstrual cycle. So you can see that its expression goes up during the menstrual cycle and in different stages of pregnancy. It goes down as pregnancy progresses. So these data were from basal plate collected from the basal plate in human pregnancy. You can see that HTR2B is most highly expressed early in pregnancy and tends to decrease its expression late in pregnancy. In contrast, so this is one of the downregulated genes. Only 9% of genes in this data set are downregulated during pregnancy. So HTR2B is one of these genes which is downregulated, and only 8% are upregulated. Most genes in this data set, 20% of them, don't change its expression. So it's not like HTR2B is like all of the other genes because its expression is going down. It's unlike the other genes because its expression is going down. Because of this, we think that HTR2B has all the hallmarks of a functionally important gene in human decidual stromal cells. And that suggests that these cells might then be responsive to serotonin, which raises the question, are human decidual stromal cells actually responsive to serotonin? So we did a relatively simple experiment. We noticed that in addition to the serotonin receptor pathways that were enriched, CAMP-mediated signaling, that pathway was also enriched. And we know that CAMP signaling plays a really important role in the evolution of, it actually plays an important role in the evolution of pregnancy. It plays a really important role in the decidualization of endometrial stromal fibroblasts into decidual stromal cells, right? We can induce that decidualization process in the lab by adding progesterone to the cells and by adding CAMP directly to the cells. So we know that CAMP signaling plays a really important role in decidualization. We wanted to explore this a little further. So we did a relatively simple experiment and asked, is there a connection between serotonin and CAMP-mediated signaling in endometrial stromal cells and decidual stromal cells? So what we did was we took a vector which has a luciferase reporter gene we took a vector that drives luciferase reporter expression in response to CAMP response elements. So there's 1, 2, 3, 4, 5 repeats of the CAMP response element upstream of the luciferase reporter gene. And we transfected that that vector into human decidual stromal cells. And then we add serotonin, wait some amount of time, and quantify luminescence expression. And what that allows us to do is see if CAMP signaling is increased or decreased in decidual stromal cells in response to serotonin. And we found that, yes, human decidual stromal cells are responsive to serotonin. So the data look like this. Here are control cells, which we have not added serotonin to, but that do have the luciferase reporter vector. If we add a little bit of serotonin, then there's a significant decrease in the amount of CAMP. So a little bit of serotonin inhibits CAMP signaling. But if we add a lot, here 200 or 1,000 micromolar dose of serotonin, we increase the amount of CAMP signaling, and sort of the dose response seems to max out, right? So there doesn't seem to be a big difference if you add 200 or 1,000 micromolar. So a little bit of serotonin decreases CAMP signaling. A lot of serotonin increases CAMP signaling. So that tells us that human decidual stromal cells have a dose-dependent response to serotonin, which as far as we know is one of the first examples demonstrating that human endometrial stromal cells are responsive to a neuroactive thing, right? So it's responsive to a hormone that we normally think is being important in brain biology, not pregnancy biology. And with that, I'll just finish up with some thoughts about the recruitment of HTR2B into stromal cells. So HTR2B is only expressed by a subpopulation of decidual stromal cells, and it's progesterone responsive. So not all decidual stromal cells express it, just some do. As far as we can tell, it's the only serotonin receptor expressed in human ESFs or DSCs, and serotonin treatment can induce or inhibit CAMP signaling depending on the dose. And these data suggest that serotonin may play a role in decidualization, pregnancy, and maternal-fetal communication. And we think it plays a role in maternal-fetal communication because the placenta produces a whole lot of serotonin. So the placenta is a source of serotonin during pregnancy. But this is like the hand-wavy bit. It turns out that a bunch of early papers in the 60s and 70s actually have suggested that serotonin plays a role in parturition timing, and that's because serotonin levels go up throughout pregnancy. Whether it does or doesn't, who knows? These are very early experiments, and this was more of a proof of principle. So we're doing experiments now to figure out whether it's true that serotonin levels go up throughout gestation and whether and what kind of effects that might have on stromal cells. And now for more general conclusions. So it's clear that mammalian pregnancy is a mixture of both conserved traits that are common to all or most species of mammal and derived traits that are unique to one or a few species. While some of those traits may be conserved, things like pregnancy and implantation and parturition, even though those sort of phenotypes are conserved, the underlying genes and pathways responsible for those traits can vary by species. This is a process that's sometimes called developmental systems drift, right? So there are 1,500 genes that change expression in the human lineage. That's a lot. And we haven't even tallied up the number that are different between human and mouse, right? So that's 3,200 genes that are different between human and mouse. That's a lot of room for species-specific differences in both phenotypes and the underlying genotypes. So because of this drift in the genetic architecture of conserved and divergent traits between species, we have to be careful about translating findings from one species into another, right? So even though the phenotype might be conserved, the underlying genes that are responsible for that phenotype might be different. And that's to say nothing about whether the phenotypes are different between the species. Evolutionary biologists like to wave their hands and tell stories. So we think that evolutionary analyses like this can help us identify these sort of interesting gene sets that might be responsible for these phenotypic differences between species. But list making can only get us so far. At some point, you actually have to start doing the experiments. It's just that it's really hard to do these kinds of experiments in humans, as you all know. So one way we can get around that is, well, we need to start functionally characterizing these interesting genes in cellular and molecular studies to understand their roles in pregnancy, and in particular to understand their roles in human pregnancy. One of the ways that we're doing this is to use organoid models of the human maternal fetal interface. So in this picture, I'm just showing you one of the human glandular organoids that we've got going in the lab, right? So it's really difficult to study these things in humans, but we have organoids now, and we can try to approximate as best we can the maternal fetal interface using an organoid system. And finally, if we want to reference any of these discoveries that we make using evolutionary biology to aspects of human health and disease, at some point we're going to have to try to relate that back to the causes of human disease, which is going to be genetic variation between individuals. So at some point we need to reference human studies, both functional and genetic, to determine the role that these genes might play in human adverse outcomes and in human biology in general, which means associating these gene expression differences we find between species with things like human GWAS studies to say, okay, here's a set of genes which look like they have human-specific gene expression patterns. Are they identified or are they enriched for GWAS hits that are associated with adverse pregnancy outcomes or any other human-specific trait? With that, I will thank the people who did the work. It was mostly Mirna, Katie, and Mavendra, a bunch of people over the years who've contributed a lot to these studies, including Gunter. You for listening, and then people who give us money. And with that, I'll take questions if there are any.